We'll start again.
Is this the right way? It's the opposite way?
My needy children. Just kidding.
This is already going well. Okay. Microphone is on. Okay. I did not know it was impossible, so I did it, by Mark Twain. In the field of biomedical engineering, or any science field in particular, trial and error is often the only way to make advancements and discoveries. For example, penicillin, we all know it, is a revolutionary antibiotic. It was discovered solely because Alexander Fleming left his lab over summer vacation and found that there was mold that staph didn't penetrate. That's how that was found. Saccharin is the world's oldest artificial sweetener. That was found when Fallberg in 1879 got some chemicals on his hands, he forgot to wash his hands, then he had lunch and found his bread was unusually sweet. The point of these examples isn't that sanitation isn't necessary, it's that there's endless potential and room to grow. Revolutionary ideas are created on accident really often, and all things are deemed impossible until someone does it. This idea is especially apparent in the science field, where experimentation is the basis for everything. So before I begin, I want to share some things about myself. I'm a senior at Frisco High School. When I'm not at the lab at UT Southwestern, I'm at work. I'm also president of student council. That takes up a lot of my time as well. And I also go to church quite often. I do like to hang out with friends, but it's harder to balance schoolwork with social life. So from freshman to senior year, I've taken a lot of science classes. I've taken medical terminology, health science, anatomy and physiology, AP biology. I double blocked physics and chemistry my sophomore year, environmental science, large and small animal management, and I did a little bit of vet tech as well. So I did a heavy emphasis on science, so I'll graduate with like 7 or 8 science credit, which is a lot. But I've always been drawn to the science field and the medical field in general. I tried a lot of different fields, and I had like a brief interest in marine biology, physics, chemistry. But I really loved AP Bio my junior year, which is why I decided to do biomedical engineering this year in ISM. So why I chose the topic. I chose biomedical engineering this year. Originally, I had an interest in zoology when I joined ISM, but I attended a biology convention over microbiology and genetics. My interest was sparked in this field, so I changed my topic to biomedical engineering. From AP Bio, I always really enjoyed culturing, I enjoyed viruses, and I enjoyed lab experimentation aspects of it, so I hope that ISM would help me bring me insight into that and maybe get some hands-on experience, which it definitely did. So, what is biomedical engineering? Biomedical engineering is the application of engineering principles and design concepts to medicine and biology for healthcare purposes. In layman's terms, it's an incorporation of a lot of different sciences to help the healthcare field, simply put. So my first semester of ISM, the very first assignment that we had was the career outlook assessment. It was for us to learn about our fields in every aspect, and as well as practice how to write an assessment, which we'll be doing for the rest of the year. I learned a lot about the field of engineering, which was solidified by my interviews, which I'll talk about later. I learned that it was a very broad and a diverse field, and it's still relatively new and growing. There's a lot of different problems and obstacles for biomedical engineering majors. The curriculum is really general, and it makes it hard for them to fit in anywhere. Because nothing is very specified, they learn a little bit of everything. For research assessments, I did a couple of them, but I mostly moved straight into interview assessments. But among the assessments I did one about stem cells and gastroenterology, how they're not being funded, but they're very useful in helping transplants and organs to regrow. I did one about robotics, so it was a cast where amputees could use them, and it would be more form-fitting and free-moving to cater to them. And then the last one I did, I believe, was about yeast evolution. I did this after I started visiting my mentor. So this one was interesting. It was about an experiment over yeast evolving that was conducted at Harvard University by Michael Desai. And it helped me understand how they were approaching their experiment, because my final product is also about yeast evolution, but they had a different take on it. They approached differently, and they put them all in the same setting and saw how they mutated with different yeast types. And we're doing the same yeast types in different environmental settings. So it's just a different approach, and that was interesting to see. For interviews, I did 8 or 9 in the first couple of weeks of ISM, so I did I did a lot. I interviewed a lot of different people because I found that there was a lot of different career paths in biomedical engineering. So mostly I did them to get a primary source about the field, and I found several different aspects of it. I found that there were a lot of careers it could lead into. It's a very large career field, very diverse, and a wide range. And I interviewed plastic surgeons, radar engineers, computer engineers, research specialists, robotics professors, head of mechanical engineering department at SMU, a lot of different people. And something that they all had in common was that they all recommended I major in something specialized, specialized engineering, so that I would have like a specific curriculum and I would know exactly what I wanted to go into before pursuing biomedical engineering in probably graduate school. So that was something interesting that I learned. My mentorship. My mentor is Dr. Erdal Toprak. He, of course, was my first choice. I reached out to him for a mentorship because I really enjoyed how much time he spent in the lab, how hands-on he was, even though he was a professor, and I hoped I could shadow him. And I got to do a lot more than just shadow him, which is awesome. So quick bio, he works at UT Southwestern in the Green Center Systems for Biology. He has a PhD in computational biology and biophysics, and he's currently studying antibiotic resistance and bacterial evolution, which is what this whole final product is in general, and what I've been studying as well. And then one of his projects was the Morbidostat, which is a robot that kind of manipulates drug concentrations in bacteria, so that you don't have to do any hands-on work. It's kind of a computer that does the work for you and adjusts the drug concentration depending on how the bacteria are evolving to become immune to them. Here's a picture of me and my mentor. It's a really bad one, next to a robot. Here's some pictures of the lab. I took these my last mentor visit because I wanted everyone else to see what the lab looked like. It's very different, and it's very messy, and there's a lot of different chemicals and things laying around, which is cool because you don't see these on a normal day-to-day basis. Some highlights of the mentorship was, first of all, seeing the lab equipment again. I never saw anything like huge centrifuges and stuff like that we were never allowed to use as in AP biology. We used Q-tips and other small pipettes. So just seeing the lab equipment, seeing different chemicals like chloroform and stuff like that in person, that was really cool, and I had like the initial excitement of seeing it and then being able to use it. Of course, being offered a student internship, that was really, really amazing. That was a really nice opportunity that I got. That was definitely a highlight of my mentorship, just meeting the right people and being able to work hands-on in the lab. The freedom given in the lab, how I was able to work by myself and have all these learning experiences, but as well as I could ask any of the graduate students questions, ask them what they were doing, and then the patience that they would have with me. I could track down my mentor and ask him, exactly what was going on, and he would take the time to explain to me and even like draw a picture for me. But I truly saw how much time and effort and work went into each experiment. They would often be there overnight, like days upon days, and it was a lot of dedication that went into each and everything that they did. So some learning highlights, some things that I learned on a mentor visit. These are only a couple of the things that I learned, but of course it would take too long if I went through everything. One of the things I learned is P1 transduction. This is an experiment done on E. coli to take out a gene. It's gene transplantation. You take out a gene in E. coli and you replace it with the one you want to make it resistant to something. So there are 4,300 genes in E. coli, and a university in Japan took out each one of them, and they sell them separately for like $10 each, and found that there's only 300 necessary cells out of 4,300 that they need to live. So if you're trying to implant a gene for kanamycin resistance, which is an antibiotic, you use a P1 phage, which is a virus, and you implant the resistant gene in the virus, and you implant the virus in the cell, and then the virus will infect the cell, take some of the cell's DNA, release some of its DNA, and then hopefully the cell will take on the kanamycin resistance gene, and then you'll use the virus to infect another cell, and so on, spreading the DNA to the desired gene. And then you'll place all the cells in a solution of kanamycin and see which one lives, which one dies, and then the one that lives successfully got the gene. So it's pretty efficient. Yeast, which is what my final product is over, it's called Saccharomyces cerevisiae. It's budding yeast. I did a lot of research on it because I wanted to fully understand what was going on at all times. I wanted some background information. I just didn't want to go into it blindly, so I did a lot of research on it. It's budding yeast. It's the most common yeast in lab experimentation. It's the only yeast with Berkeley bodies present. It can be haploid or diploid. Something I found interesting was that haploids will die under high stress, and they're asexual. And diploids under high stress will actually sporulate, and they can double their population in like 100 minutes. So it has a very short generation time. It's easily cultured, so it's easy to work with in a lab. It divides with meiosis, so it's very easy to use in sexual genetics research, which is what we're doing after this experiment, my final product. And it shares actually the cell structure of plants and animals without non-coding DNA. PCR is polymerase chain reaction. It's a fundamental process in biology I learned about. We briefly touched on it in AP biology, but never got to see it hands-on. It shows a mutation, and it's used to test the reaction and the growth of mutation to see if the bacteria grow resistance, in my case. So you do the chain reaction about 30 times with DNA polymerase, just replicating the gene ATGC, and see if the bacteria will mutate or not. And if it does, then it grew resistance, and if not, then you try it on a different plasmid. 9-6 wells are something I've never seen before in biology. They're tiny plates, A through H on top, 1 through 12 on the bottom, and they're filled with amino acids, in our case, and you never use the four corners, never use outer edges. You skip in between to avoid contamination. They're how you create master plates. They're what you use in the robot to read DNA growth and create graphs as well. These taught me that precision is really important, and a lot of times we would mess up all the 9-6 wells because you place an amino acid in the wrong well because they're very tiny. So that taught all the patients as well. How to make plasmids, I also learned. It's actually a kit called the Kyogen kit, and it comes with everything that you need: buffers, bacteria, which is cool because I thought that kits we only use in student learning settings, but scientists actually use them too, and it's pretty efficient. Centrifuging, we learned briefly about in biology as well. It's used to separate by gravitation and density, but something cool I learned when I got to use these was that they come in different sizes, and it depends on the powerfulness of them, depends on the size. So there are ones that are literally the size of like half this classroom. They're so big. And they have different ranges of power. So those are just a few things that I learned during my time in the lab. So my original work. My original work was a protocol document for doctors to prescribe antibiotics safely and effectively to combat the growing antibiotic resistance. I wanted to utilize this original work and expand my knowledge upon antibiotics. I wanted to create a policy for doctors to prescribe antibiotics safely, and the healthcare field to grow awareness for the growing antibiotic resistance. So I just wanted everyone to know about that there was a global threat happening, that it was potentially very serious, and I found that the root of the problem was physicians, and I wanted to answer the question why antibiotic resistance was rapidly growing. So some obstacles, some difficulties I found in approaching this original work was deciding the approach on how I would direct this towards professionals, knowing my age and my limited amount of knowledge. How could I reach out to medical professionals who had gone through training and school? I had the self-awareness to know that I didn't have the necessary knowledge that could tell them what to do, but I thought it was worth a try to give them some recommendations, even if it potentially just spread awareness, potentially just saved a couple of lives. So the research I did a lot of research because, again, I had a limited amount of knowledge on this topic. I just know that I wanted to learn more about it and do this. So some research I did was, I first researched about antibiotics. So specific antibiotics, new antibiotics, the types of viruses that develop resistance. I looked for common factors in both the antibiotics and viruses, why the antibiotics grew resistance, what kind of factors that they had. I found that they were being heavily prescribed with not enough reason to be prescribed, and that's why most of them had grown resistance. For viruses, it was either the form, the structure, their function, how readily available they were in the environment. Those are some common factors that I found. So I scoured a lot of articles. I read each one twice, and I had hundreds of papers. It was a lot of work, and found some great study places to do this. And the ones I found I could use in my original work after I read them each twice, I kept those, I highlighted them, and I annotated them, and I put them in a binder so I could organize it later into a general outline. So I started with that, antibiotics, viruses, types of viruses that gain resistance. And then I moved to mutations, how the viruses mutate to become and evolve resistant, the effects of antibiotic resistance globally, how it affected people, the statistics, how many people it was killing, and the potential seriousness of it, and the types of antibiotics that led to resistance. So I also researched how to make a protocol, because I was eventually making a protocol for doctors to prescribe antibiotics, so I needed to know how to outline it in order for it to seem professional and correct, how to create regulations. I researched where the resistance came from, whether it came from a specific gene, a supervirus, and how they occurred, how they evolved, where they evolved. Overwhelmingly, I found haphazard prescriptions to be a very large common factor in each resistant virus or antibiotic. Example would be vancomycin, which is a drug. It's only supposed to be used in life-threatening situations. However, it's being prescribed very haphazardly again, very casually, and so it's grown a lot of resistance. Vancomycin isn't as effective anymore. So this very rare drug that could have saved a lot of lives is no longer effective. What I found is that each article that I read branched off into 10 more articles, because I had so many questions after reading one article. There'll be a lot of things I didn't know, a lot of terms I didn't know how to define. So I would research more on that, and it would lead to 10 more articles I had to read. So it was a very cyclical research journey. I use a lot of databases for my sources. I use a lot of scientific journals and a Google Scholar, which is where I got a lot of my articles. So after I was done reading all these articles, there's a lot of articles, so many. I found a couple of topic sentences between them, some things that I wanted to elaborate on. After I annotated them and highlighted things I wanted to include, I organized those into supporting information for each topic sentence and all the evidence in the articles. And then I created a connection. I linked each one I wrote. I decided how I wanted to organize it using annotations. I knew I definitely wanted to include an introduction, a conclusion, and a transition, just so I have more reasoning, more reliability behind what I was saying. And even if it was just to potentially make aware like what their role was as physicians in this growing antibiotic resistance, an introduction, some statistics to show them how serious it was, and a conclusion to wrap up what was being done already and what was already in place to combat it. So I did an outline of the protocol. It was very rough bullet point format, where I listed out the evidence, the reasoning, and the rule that I wanted to be in place. And then I put evidence for each number so it would be more reliable, like an example of an antibiotic that had grown resistance because of this reason, or a statistic I found that people were being negatively affected because physicians had done this. I had a works cited page, and yeah, here is my protocol. There's 20 of them total. The introduction is just I introduced penicillin, how that grew resistance. It's the most common one. Thought it'd be very familiar. The threat, how it's killing many people a year, and how it's affecting even the economy. I found that it was resistance was in tuberculosis, malaria, staph, gonorrhea, pneumonia, septicemia. A lot of common diseases were growing antibiotic resistance, which would make them a lot more deadly in the future. So a common cold could potentially kill someone very easily. So I mentioned that there were potential actions being taken, but then that they wouldn't grow unless physicians cooperated. And then my transition was about the role of physicians, why they were so important, and why their cooperation was so important, and why prescribing antibiotics properly could save a lot of lives. And then here's my protocol. Go through. The italicized font is the either an elaboration or the evidence. And in my conclusion, I just talked about, again, I emphasized how the role of physician was very important in this, not to prescribe haphazardly. And I also talked about global cooperation, the other things that were at work that the physicians could work with in order to combat growing antibiotic resistance, things like Teixobactin and Bill Gates Foundation. They get my excited. Okay, and then my final product. My final product is a yeast evolution experiment. It's an experiment to see how they develop in different environments. So there are 51 conditions. There's 50 amino acid conditions and then YPD. And there are 4 yeast replicates. So there's 2 alpha and 2A. Alpha and A just mean it's essentially a gender to yeast, so they can mate. So 51 conditions, 4 replicates, 204 cultures a day. So I was labeling test tubes 204 times a day, and we were transferring yeast 204 times a day. That was a lot. That was really tedious, but that part of the experiment is over, and that's just the setup, which is also really important. So for an example, you have ALA 1 through 4, which is an amino acid. You have 51, so it's ALA, ARG, like all the way up to uracil, valine. So you have ALA 1 through 4 in its amino acid media. The yeast will consume the media overnight, and then you transfer it to the new media the next day, so it can continue to live. Then you have delta 1 through 4 of each amino acid as well, which is everything but that amino acid. So essentially for each amino acid you have eight pairs. You have all of those amino acids, and you have SC, which is all of the amino acids, none, which is none of the amino acids, and then YPD, which has no delta. And at the end of the experiment, you measure how the yeast adapted to the different environments. Sometimes the yeast, there was a lot of problems and obstacles in the very beginning because we had never done this before, and no one had ever done this before. So it was a kind of a trial and error type of deal. So the yeast might not grow in a certain media. Tryptophan 1, we had a lot of problems with. The yeast just wasn't growing exponentially at all. And you can tell these are grown because the amino acid test tubes, they don't get cloudy, which means that the yeast isn't replicating, and the amino acid is still there, not consumed. And then eventually, tryptophan 1 turned pink, and all of the test tubes turned pink, and we weren't sure why. We still don't know why, they just turned pink. So we haven't been able to figure that out yet, which we'll do after this whole experiment is over. So we just kind of canceled out tryptophan 1. We found that it wasn't really working. So later on, we're going to use what we got from this data and this data analysis to mate the yeast together and have a mating experiment, which is a separate experiment entirely. If we see that the yeast evolved to grow resistant to a certain type of gene, such as alanine or arginine, we'll mate the alpha and the A yeast who grew resistant to 2 different types of antibiotics and see if the yeast offspring develop a resistance to one of the antibiotics, both of them, or even like neither of them. Some other aspects of the experiment was the robot, which was also in the picture earlier. Basically, the robot is used to check fluorescence in the yeast, and this also shows the growth rate, how they're growing, and we check these in the 9-6 well. So data analysis is another aspect of it. These are just graphs. There are 204 graphs, so there's a lot. We use MATLAB to do them. There's a lot of coding involved as well. That's something interesting that I had to learn. And then the transfer process, which is just transferring the yeast from test tube to test tube to new media. This is another tedious aspect. Not a bad aspect necessarily, but just like a very laborious aspect. Because you have to put the amino acid in each test tube, transfer the yeast 204 times to each different test tube, organize them into their respective amino acids, and then label them 1 through 4, and then delta 1 through 4. So that was cool. So the development of the product, the conception, here's some pictures. This is the test tubes that I labeled, 204 of them, and then the ones wrapped in foil are the amino acid media. We have wrapped them in foil so they're not exposed to light, which would kill the amino acids. And then here is the fluorescent bacteria, or the yeast. It goes under a black light. The one that you see on the picture is the one I have right here. I'll pass it around. don't open it, but like I'll pass it around. Yep, that is Saccharomyces cerevisiae. It's budding yeast. It's M cherry, which is why it glows. So under black light it'll glow, but not just under regular light. And you use special infrared glasses to see them. Yeah. So for conception of the product, my mentor pitched it to me. He said, I'm working on an experiment and I would really like you to join me. And so I was really, really excited to work hands-on in a lab like this. So I really jumped at the chance to participate in one of these experiments. The completion, it won't be completed in time for final presentation night, or even in June it won't be completed. So it'll probably go over the summer, especially because it goes hands-on with the maiden experiment. So I'm excited to keep participating in that and keep working on that as well. And my conclusion. For how I've grown from taking ISM, I learned so much about everything, not only about my topic, which is microbiology and biomedical engineering, and about my career, but about myself as well. It was as much work as I was warned about from last year's students. There's a lot of deadlines, but it was just as rewarding as they told me it would be as well. It was a lot of responsibility, and there were, again, a lot of deadlines, but they helped me a lot. It was taught me a lot about professionalism and growth. I really learned how to communicate with adults and professionals in a professional setting, dressed casual, like business professional. Learned how to prepare speeches, talk for 30 minutes at a time, and how to prepare even like invitations were something new that I had to prepare. Again, keeping a journal of everything that I did in the lab, all the things that I learned, all the things I did that day, so I could keep a mentor log as well. And it was really helpful for me to keep a scientific journal, so I could, even creating this presentation, it was pretty useful to me. I learned how to dress professionally, just in all aspects, even like going on interviews, even though it was slightly uncomfortable at first, meeting new people, putting yourself out there. It was a really nice thing to learn, having a firm handshake and how important that was. I also learned the importance of asking questions, even in interviews, just asking everything that you wanted to know, because the only way they would answer them, the only way they would know what you're thinking is if you asked questions. For my mentor, especially in the lab, if I have a question about anything, I'm no longer hesitant to ask him, even though it seems like I'm bothering him, if I'm like pestering him. It's really important for me in a lab setting to ask questions so I know what's going on at all times. I know like what's the reasoning behind what we're doing, especially with a lot of experiments that are very complicated, that have different aspects of it, things like my final product. So I learned that, and I learned so many other things besides that as well. And moving forward, I hope I can use these skills I learned in ISM in college, not only communicating with professionals, but because I want to go into the healthcare field, I also do want to have an internship, maybe doing research. So hopefully the experiences I had can also help me determine the exact career field I want to take. I plan to major in biology at UT Austin, and hopefully join the Freshman Research Institute, which will be great because I've already done some research and I've worked hands-on in a lab, so I kind of know what's going on in the correct protocol. And doing research and experimenting in ISM this year really helped me determine that I love the science field. And hopefully moving forward, I can utilize all the skills I learned, everything I got from my experience and from my mentor for a long time in the future. Lastly, I want to say thank you to everybody who came. Thank you especially to my mentor for taking me to his lab and allowing me to have these experiences. And thank you to everyone who supported me and helped me in making this year possible.